Thank you very much, and I really appreciate the opportunity to speak with you today. It's actually quite surreal for me. Probably like many of you in the crowd, I met Dr. Borlaug at a pretty young age. I was 21 years old, and oddly, and the only time it ever happened, he and I were completely alone. He walked into the cantina at El Baton on a Sunday night, just arriving. I was just a young college student, and I say that because ever since then I felt these pretty discriminating eyes, over my shoulder, and they've been there, and so it's really an opportunity to talk about work that's dear to his heart, I know, and to his beliefs 15 or so years later. And I think many of you in the crowd remember in Ciudad Obregón, 3 or 4 years ago, their first BGRI technical conference, he gave another one of those passionate, assertive, get your butts to work speeches. He he was good at that, and I am pleased to come today and talk where I think he would be proud. Ronnie stole most of my comments earlier in his introductory remarks, but I think he would be proud. I remember him commenting just from that first BGRI technical workshop, how he was encouraged or optimistic about some of the stuff he saw, some of the presentations and research he saw at that first the first session. So it's a journey from that first BGRI technical workshop and basically 4 years of the DRW project and BGRI to Beijing here. I gotta figure. Okay, thank you. I need to acknowledge this is not my talk, this is not my research, just like Dave before me. This is a collaborative effort involving many outstanding colleagues and scientists around the world. There's too many to list individually because there's several at multiple institutions, so I've just listed the organizations. And it's a quite diverse group. Work spans from very basic pathology to wearing funny Tyvek suits in the middle of a field in Africa, to some quite molecular work where we're actually, in some cases, looking at the interactions of resistance genes and how additive or how they interact, developing genetic markers, developing new resistance sources from cytogenetic stocks, and even in some cases down to what is the gene that's causing this resistance. I'm missing something. This screen. Okay, sorry. So with the title of the talk being Stocking the Breeder's Toolbox, wanted to first talk about why do we think we need a better toolbox and what's motivating this. And really the bottom line is to speed and support replacement of rust-susceptible varieties based on complex resistance globally, with an initial, of course, focus and emphasis in East Africa and Southeast Asia, but really to follow Dr. Borlaug's mission of eliminating or really getting on top of the threat of stem rust and other rust of wheat. We're trying to do that through increasing diversity of resistance and promoting durable resistance in various forms. And trying through these more basic molecular genetics and pre-breeding activities enable stewardship of resistant sources in a manner that's never before been possible. So maybe that shifty enemy that never sleeps can take some rest. So what tools are needed? First, we believe a well-surveyed resistance gene landscape. So basically knowing what our source of resistance are, where they are, what sort of pathogenic variation or virulence, avirulence there are to various sources of resistance. Looking at which genes would be most useful in different combinations in different areas of the world. Initially, and proud to report or show that we've made quite a bit of progress in this area, we feel like we needed additional useful sources of resistance to really expand the diversity that was available in breeding. We need effective gene pyramids, and sometimes that may be talking about actually recombining genes in similar chromosomal positions and be inherited as one unit, or combining genes across the genome in ways that are going to be durable. And for most of those breeding efforts, having DNA markers or supporting information that really allows you to robustly select across diverse germplasm, no matter what it is, for complex resistance and postulate or diagnose resistance in breeding germplasm around the world. And you need good germplasm in the end. Having all that information doesn't do any good if you're not delivering it in some usable form that a breeding program or breeding programs throughout the world can use. So when I say complex resistance, collaborators in the DRW project believe that can be achieved through a few different ways. One of those is deploying combinations or pyramids of effective, broadly effective resistance genes or hypersensitive response, seedling, all-stage resistance, what we have relied on many years and have caught us or made us wish we hadn't sometimes as well. As well as adult plant or slow rusting resistance, where we also need, by default, to get the appropriate level of resistance, complex additive action to combine those genes in a way that actually confers effective control. And I thank Ravi Singh for this slide. Ravi has used this many times over the years, and I found it quite effective as well. Basically, what we're going for, or one way to look at it, is a seedling resistance gene can give you any level of those resistances, from kind of MS, higher, not so strong resistance, all the way down to completely clean. And that's kind of the problem with seedling resistance genes is you can get that beautiful control with one single gene quite easily. Whereas APR breeding, you have to work quite a bit harder to get that effective level of resistance, but it is possible. Basically what we're saying or promoting or believe in is that we need to be focused down there on that bottom line. It comes down to the bottom line, which is having complex 4 to 5 APR genes or three or more race-specific genes pyramided to really work towards durable control of rust. And so to do that, you need to have a lot of genes, and you need to know a lot about them and be able to combine them, even though with a race-specific gene, you may not be able to see one behind the other. You need to have tools to be able to do that. If we look back essentially at our first BGRI technical workshop, or about 4 years ago, this is kind of what the sort of resistance gene landscape looked like. The number of genes on this map, and these are specifically that have some effectiveness or documented effectiveness against UG99 group of races. There are 21 there, and of those 21, several were inaccessible because they were on alien translocations in forms that had never been usefully deployed due to linkage drag or other problems. And other genes on this list simply, they can be effective or useful in some ways, but they may not be the best genes. So we're somewhat limited. I kind of like to think of this as with 21, it's a haploid, where it's just one part of the puzzle we need to put together. And it's been quite encouraging that the picture changes quite a lot as you move forward just 4 years. So if I back and forth, you can see it's quite a change. Actually, there's about 48 different things on there. So we've gotten up to a diploid again. We've formed an embryo that's kind of the place that I think we need to grow from now. I've highlighted in green adult plant resistance loci that are confirmed to have effect in reducing UG99 and I think as well as other races levels of resistance. In red are those that are derived from alien species or wild relatives that show less recombination in the wheat genome, and in black are those that are just of common wheat origin. So there's a lot to take in. There's a lot of new sources of resistance, a lot of information there. I'm going to go through a few of those. The first one that is still one that we rely on and is the backbone of a lot of resistance around the world is SR2. That story's been told a lot, and I'll kind of just focus a little bit on a few genes there's been significant changes in the past few years. Just starting with things like SR12 and 15, these are genes that are based on enough work over the past 3 or 4 years, are consistently showing some moderate effects, contributing to UG99 resistance in multiple genetic backgrounds. These are things we didn't really know 4 years ago. And those are potentially useful because they're already in common wheat germplasm, potentially in quite a bit of materials around the world. Other genes, for example, SR32, it was in an Aegilops speltoides translocation that had linkage drag, and that's since been reduced. There's markers for these. Similar story for SR22. In the cases of genes like SR33 and 35, those have actually been cloned, and we now know the identity of that resistance gene. For each of these, there's been significant developments. I do encourage you, I've tried in the paper accompanying this presentation to outline where you can get more information about a particular source of resistance that will go in more detail in the pathology or the status of markers and germplasm. The story continues. We get more and more sources. Actually, I guess when we started with this DRW project in 2007 or 8, we were up to about SR46. That was and so we've since progressed up to SR57 now. So that's about 11 new firmly designated genes in that time. Many of these are actually from common wheat origin, some are from wild species. Some are already produced on reduced translocations where linkage drag is eliminated, or we have very good markers if they're from common wheat origin. And as I mentioned on the previous graphic or map about the sources of resistance, really documenting the contribution of these broad spectrum APR loci has been a significant accomplishment, and it's actually resulted in additional designations of like the LR67 YR46 locus on chromosome 4D as SR55 as well. Similar story for LR34 and probably soon to be changed or reported for LR46. In each case, careful detailed studies have showed that they are contributing to adult plant resistance to stem rust. In addition to those formally designated genes that were up to in the 50s, there's a number of genes that, based on all data to date, are unique and potentially effective, or not potentially, but are effective against UG99 and represent new source of resistance. There's 4 or 3 listed on this slide. Some coming from species such as Aegilops tauschii or Triticum monococcum, but as well as genes discovered in common wheat germplasm. There are additional temporary designated genes, and if you look at posters around this hall, you'll see there's other source of resistance coming as well. So it's actually quite an exciting time. In addition, association and biparental mapping have found consistent QTL across no less than probably 6 to 10 populations, as well as large association mapping panels that are consistently showing up when tested in UG99 or East African screening nurseries. These, I think you can expect, will be designated and validated over time just due to their consistent effects they've shown so far. And in addition to all those new things, there's a lot more coming down the pipeline. Many of you in the room are involved in resistance gene discovery efforts. There are many projects now working in this area. I'm just going to go through a few examples from the DRW collaborators. Brian Stephenson and colleagues at the University of Minnesota, as well as UGen and at Tel Aviv University, have been doing a lot of work with wild Aegilops species, a lot from around the world, but specifically Israel in this case. You can see in many of the Aegilops species, you see a very high frequency of resistance indicated in the green when tested with UG99 and other group members. These, in several cases, we haven't ever transferred a resistance gene from those species, so we have reason to believe that there are new sources that are unique that are going to continue to contribute to weed improvement. So sources or projects like this are at various stages of development, from introgression into hexaploid wheat, or doing allelism tests within the actual species to try to sort out what's unique before transferring into a hexaploid background. There's also genetic mapping of those, both in the diploids or in translocation, or not just diploids, but in the wild species, and some chromosome engineering at various stages with partners across the world. There's, in addition, or through these screening efforts, various age-elapsed species, wild tetraploids, Dasypyrum villosum, have shown to have probably a number of additional resistance genes available from those gene pools. And all of those are, like I said, at variable stages of introgression or characterization. And even in some cases, Stephen Zhu has a poster back here showing association mapping just right in the tetraploids for new sources of resistance, and likely identifying 3 or 4 or 5 additional new genes, seedling resistance genes in that case. We continue, the first few years we're focused on those alien genes that were on translocations that were already well known, that were high priority and got done. That would be like SR39, SR32, SR40, SR47. There are probably no less than 5 to 8 additional alien sources that are very promising, that are being transferred. This is examples of Stephen Zhu's work, where a nice small translocation harboring an additional gene from Thenopyrum intermedium was transferred and is under validation and additional characterization right now. Marker development, it's really an exciting time. Tools just in the past 2 years for wheat genetics have changed dramatically. Sequencing, genotyping by sequencing, using 9,000 SNPs, 90,000 SNPs. These are things that we really didn't have access to even 15 months ago, and they were really accelerating the development of markers and sort of characterization of germplasm, but also identifying unique new resistance sources. This is largely, as much because the data is consolidated and standardized and allows us to warehouse it in a way that we couldn't before. When individual labs are running this type of marker or that type of marker, and you have a picture, it's pretty hard to share that at a national, international level. But by having these databases of really dense and standardized SNPs, it allows you to look across the germplasm and much more quickly identify those things that are the same or different. And it really is a promising time. For example, to help aid in sort of postulation or identification of new sources as we map genes through various means, scientists and USDA have contributed about over 160 resistance sources or stocks for 90,000 SNP genotyping. From USDA, that's Matt Rouse. About 90 of the leaf rust resistance sources from Jim Comer's program, and over 400 stripe rust resistance sources as well. And what we're finding through various studies is basically by having those donor various sources from around the world already haplotyped and knowing the markers, when we identify a gene through some mapping approach or association mapping, it allows you to kind of quickly go to what exactly. Sometimes the markers are diagnostic right away, and only that one resistance source shows up as having that allele, and so it really is speeding the process. And through the DRW project, numerous populations have been or are being genotyped by either 9,000 or 90,000 SNPs, and that will continue over the next couple of years. I already mentioned before, in some cases, the sort of relative ease of doing this type of work in wheat due to the tools we have has led to map-based cloning of so far SR33, SR35, and there are others imminent that's going to provide additional tools to combat rust maybe in the long term. One effort that I'm particularly fond of and I think is going to be rewarding is development of linkage blocks, where as you can see from the genetic maps, they're a number of chromosomes where we have several resistance genes. I would not necessarily clustered. In some cases they are, but present fairly close to each other, close enough that it makes sense to go ahead and intentionally cross, recombine, select those that instead of having the genes in repulsion, they're in coupling on the same chromosome, and probably with just one DNA marker, you can, or two DNA markers, you can bring those along together and enhance their sort of durability by protecting them together, but also do it easier. You're not having, if you're trying to pyramid 3 genes and 2 of them are already linked, that makes your job a lot easier to begin with. So active efforts, I've thrown just a few examples from the previous slide I presented, but active efforts right now are focused on chromosome 1D and 2B, where it's clear that it's going to be feasible, and we hope it'll be quite useful germplasm and genetic resources to enhance stewardship of these genes. Even newer tools that are emerging and quite exciting, things like genomic selection and actually just predicting based on genotype and existing phenotypic data what the level of resistance of a line is going to be. This is work from Jessica and Mark Sorrell's group showing the accuracy of predicting adult plant resistance to stem rust in 2 different populations that are part of her research. And on the phenotypic distributions on the bottom right, you can see that just based on genotype alone, they are skewing the proportion of resistant lines. And although it may not be as perfect or as good as phenotypic selection, it can be done quicker. It can be done not in Kenya or East Africa, where we have limited screening nurseries. And we think this is going to be, a large part of the future of resistance breeding, but wheat breeding in general. So what's all this mean? To me, just like Dave, I think there's never been a more opportune time for all of us in this room and our colleagues across the world back at various institutions to actually take all this information, embrace it, start thinking very hard about strategic deployment and responsible stewardship of these genetic resources. And it'd be a shame. I don't know if anybody's a shade tree mechanic like me, but I often lose tools pretty easily. My toolbox doesn't stay full, and I'd hate to see that happen here. There's been a lot of time, energy, excellence invested in developing a lot of these resources, and it's going to behoove all of us to use them in the best way possible. So I'd like to acknowledge a similar list as Dave, the critical support and vision and flexibility and just collaboration with the Bill and Melinda Gates Foundation and DFID that supported much of this work, Cornell University's leadership of the DRW project and scientific contributions from Cornell, as well as all the other collaborator institutions and BGRI partners. And I'd like to thank the Institute of Plant Protection for hosting this and welcoming us to China.